for the introduction, and thanks again to the organizing committee for inviting me to give this lecture. I'm sorry that I'm not able to join you in person, but it's an honor to have the opportunity to talk today about how far we can go with fMRI. And so with this talk, I hope to convince you that the blood vessels of the brain reflect neural activity and functional architecture remarkably well. Now, my lab develops technologies for fMRI, mostly high-resolution fMRI, but I wanted to use this platform to develop a thesis that by understanding the vascular influences on fMRI at a deeper level, we can extract more neuronally specific and meaningful information from our fMRI data. And so for this reason, today I'll address the question, how do brain vascular anatomy and physiology shape or influence hemodynamics and the fMRI signals? Now, as this audience knows, fMRI allows for non-invasive measurement of neural activity across the entire human brain, from cortical gray matter to subcortical gray matter and brainstem nuclei, as well as the cerebellum and even the white matter. However, all fMRI signals in use today measure neural activity indirectly by tracking the associated changes in blood flow, volume, and oxygenation that accompany neural responses, which are collectively known as the hemodynamic response. Now, because hemodynamics are viewed as being a poor surrogate of neural activity, this aspect of fMRI has often been viewed as the fundamental limitation of the technique applied to neuroscience and brain mapping. But how much of a limiting factor is this? The blood vessels of the brain are quite remarkable, and the responses are far more precise than many realize. Now, in the context of brain mapping, a related question then is, what is the resolution of fMRI, and how well can fMRI localize brain activity? Now, this is relevant for those of us who wish to map units of functional architecture, such as cortical columns, layers, brain parcels, and subnuclei, as well as fine-scale subnetwork organization in large-scale brain circuits that Randy Buckner and others have recently demonstrated. So to address this question, we should first distinguish between two concepts. We've seen that advances in MR technology provide smaller voxels and faster sampling rates, thus increasing the imaging resolution. So here's a recent example from Avery Berman from our lab showing a high imaging resolution of 0.6 millimeters isotropic fMRI at 7 Tesla, which required a lot of effort to achieve. And so with this capability, the natural question arises, is the accuracy of fMRI today really limited by voxel size or by the biological resolution of the technique, by which I mean the precision in space and in time of blood flow regulation in response to neural activity? Now, conventionally, the biological resolution is thought to be coarse in space, maybe 1 to 2 millimeters, and sluggish in time, with the hemodynamics peaking 6 seconds post-stimulus. However, our colleagues, mainly in the field of optical microscopy, have shown us that fundamentally, the biological resolution of any hemodynamics-based measure is far better than previously believed. Now, the biological resolution is thought to be limited by the spacing and timing of the control structures of blood flow, which were presumed to be the smooth muscle cells that surround arterioles, and maybe these are about 1 millimeter apart. However, mounting evidence suggests that pericyte cells that have been known for a long time to wrap around capillaries are also capable of controlling capillary diameter. So here you can see a movie of pericytes in action. So you can see here a single capillary with individual red blood cells. In the middle is an electrode attached to a pericyte. And when the electrode stimulates that pericyte, it constricts, showing that it's contractile and we can regulate diameter. Pretty cool. But perhaps what's more amazing is that, and this movie loops in case you missed it, that just after the stimulated pericyte in the middle constricts, the neighboring upstream and downstream pericytes, indicated by red arrows, also constrict. So there's some coordination among these pericytes, and we know that there is signaling or communication along the endothelial cells that line the vessel walls. So this tells us that there is machinery in place for very focal blood flow regulation. Now, the bad news may be that there's some coupling between the diameters of multiple capillary locations, which may constrain or limit the degrees of freedom of blood flow regulation. Also, the layout and connections of the capillary network will also affect how blood flow is regulated. And I'll consider this in detail later in the presentation. Now, these remarkable data were acquired in vitro in excised retina, where the blood pressure and flow aren't really at physiological levels. However, the same group revisited this years later and showed that pericytes are able to actively control capillary diameter in vivo as well. Hopefully you can see them dilating in this movie, and this capillary dilation precedes the upstream arteriolar dilation, so the dilation seen at the capillaries is not a passive response to upstream flow or pressure changes. Now, while there's still a debate over whether the cells observed in this study were on capillaries or perhaps pre-capillary arterioles, and there's some disagreement still over whether all pericytes are contractile. Either way, these data show that there are mechanisms in place for very tight blood flow regulation at fine spatial scales. Now, briefly, while these data were all taken from small animal models, there is recent evidence
Can you hear me now? Okay, I'm going to shout. Okay, we've tried to slow the video down, but I don't think we can. Is the sound quality better? I think I could, it seemed better as it went along. Do you guys feel the same or no?
Okay.
Okay, thank you.
Please continue, yes.
It's based on microscopy data of excised tissue removed during brain surgery that pericytes play a similar role in human brain as well. And so this suggests that the biological spatial resolution of hemodynamics-based human fMRI may also be quite high. So now considering temporal resolution, in this study conducted by Anna DeVore's lab, they measured in mouse S1 calcium dynamics in astrocytes concurrently with neighboring single blood vessel dilations in response to 4-PA stimulation, and found that while the astrocytes activate about 3 seconds post-stimulus, the vascular activation began on average at 0.7 seconds post-stimulus, and sometimes faster. Now, not only do these data question the role that astrocytes play in neurovascular coupling, but they also teach us that the smallest vessels, if we can see them, respond very rapidly to the stimuli, again suggesting potential for perhaps surprisingly tight blood flow regulation and a high biological temporal resolution. And so if we consider these results together, they suggest that the biological resolution is likely to be higher than even what we can achieve with modern fMRI. Now, this motivates us to develop technologies that enable smaller voxels and faster acquisitions, although today it's still unclear what the ultimate biological resolution is, and it's likely to vary across brain areas. It appears to be tighter than 0.6 millimeters, so even smaller voxels would be put to good use. However, our ability to localize neural activity with fMRI is likely influenced by the vascular anatomy and physiology, as well as its structure. And so what makes the biological resolution difficult to measure is that the brain's vascular anatomy closely reflects its functional anatomy. The so-called angioarchitecture of the brain exhibits clear vascular layers that match the cell layers, as shown here. Vascular layer 3, having high capillary density, contains cell layer 4. However, there's not really a one-to-one mapping between vascular and neuronal layers. We also see that different cortical areas exhibit distinct angioarchitectures. This can be seen here, so just to orient you, this is the human visual cortex with a calcarine sulcus here and the white matter here, and we can clearly see that this band of high capillary density in the middle of the cortical gray matter drops off abruptly at the V1, V2 border. Now, this can also be seen at the border between primary motor and sensory cortex, M1 and S1 here. Now, while human brain mappers know well the classical brain parcellations of the cerebral cortex based on architectonics derived from laminar differences in cell size or density or myelin content, as seen here in the famous atlases of Brodmann, the Votes, Flechsig, and others, what may be less known is that similar parcellations have been proposed, such as this angioarchitectonic atlas from Pfeiffer, who have trained under Flechsig. So here you can see a photograph of the angioarchitectonic fields painted onto a wooden model from the macaque cerebral cortex in lateral and medial views. Now, many of the cortical areas of Brodmann are also seen in this angioarchitectonic atlas. So this tells us also that there's a close relationship between functional brain regions and their microvasculature. Now, this tight relationship extends to other brain regions, such as the thalamus, as shown in this beautiful example, where one can see in these vascular stains the 6 distinct cell layers of the lateral geniculate nucleus recapitulated in the microvascular density, as well as nuclei of the brainstem. So here we see the LGN again, but also the red nucleus in the mammillary body down here. Also, the inferior colliculus, which is not seen in the slice, really pops out as well. And so the vasculature is adapted to the function of the brain. And while vascular density resembles cell density, perhaps unsurprisingly, it's more strongly correlated with patterns of neuronal metabolism, as shown here. So not only does this mean that the vasculature is finely tuned to deliver energy to the brain precisely where it's needed, but it also tells us that the brain's functional architecture is reflected in its vascular architecture. So therefore, it may be possible to mistake fMRI activation patterns that really just reflect the vasculature for patterns of neural activity. And knowing where the control structures are is not enough, since hemodynamics are also shaped by vascular topology and geometry, and I'll revisit this at several points during this presentation. And so after this motivation, here's an outline of the topics I'll be discussing. First, I'll present anatomically informed cortical depth analyses of columnar fMRI data and ask the question, can knowledge of the vasculature help us to improve the sensitivity or specificity of the BOLD response? Next, I'll discuss tissue orientation dependence of BOLD fMRI in the gray matter and white matter, and ask, does a coupling of vascular and tissue geometries cause detection biases in fMRI? So then I'll present our work on physiological resting state fMRI networks, and here ask, can we differentiate between vascular networks and neuronal networks in our fMRI data, which I'll define. So I'll also show some recent work on biophysical models of fMRI responses and ask, can realistic vascular models help us to estimate this biological resolution? And I'll conclude by showing some fast fMRI results tracking delta band frequency oscillations in BOLD and pose the question, how sluggish really is the BOLD response? Okay, so first, anatomically informed analyses. So the starting point here is that there is a hierarchy of vascular anatomy in the cerebral cortex, which can be appreciated from these microscopy data from monkey V1. So on the surface of the cortex, we have these large pial veins that drain deoxygenated blood from large cortical domains, and then the ascending venules that run perpendicularly across the cortical layers, and here in the parenchyma, the seemingly randomly oriented capillary bed. So the idea is that as our fMRI voxels become smaller, we can exploit this vascular organization and, for example, sample exclusively from voxels at the capillary bed where the coupling between the hemodynamics and the neural activity is tightest, while at the same time we can discard voxels sampling from the large veins that carry non-specific BOLD responses. So to test this, we utilize the well-known retinotopic mapping in the human primary visual cortex to generate a visual stimulus that would elicit a ground truth neuronal activation pattern in the shape of the letter M, as shown here. And then we sampled our small fMRI voxels across the cortical depths to test how the activation pattern changed when sampling either close to or away from the large pial veins. So here is the activation pattern on the inflated cortical surface. You can see that it's tough to see the M activation tucked inside the calcareous sulcus in the original surface reconstruction, but if we inflate the surface reconstruction, we can better see the activation pattern. So we can use knowledge of what the spatial pattern of neural activity is supposed to look like to evaluate our fMRI data. Now, if we sample close to the white matter, the spatial fidelity of the pattern is quite good. But as we progressively sample closer and closer to the pial surface, the spatial pattern breaks down systematically, but I think in an informative way. So for example, we see this artifact at the pial surface that may be a vein that's cutting off the top corner of the M. So this tells us that the biological resolution of fMRI is complicated and does not appear to be a simple Gaussian point spread. And the pattern observed at the pial surface is not simply a smoothed version of the pattern that we see at the white matter surface. This is because our fMRI voxels are small and therefore sensitive to the local details of the vascular anatomy. Now, these are old data, and I'm including these here today because this was a fun experiment that showed us in a concrete way something that many had suspected about the pial veins. However, when I first presented these results about 13 years ago now, the community had a hard time accepting that the biological resolution of fMRI was high enough to even see this pattern based on the understanding at the time. And so we've come a long way since then. So we can look at the activation strength across depths as well. So here's the activation strength at the white surface, and here it is at the pial surface. We see that the percent signal change is about twice as high near those large veins that it is at the white matter. So if we consider this alongside the activation maps, together these plots tell us that, unfortunately, the sensitivity of BOLD is highest exactly where the specificity is lowest, at the pial surface. So this trade-off means that for columnar systems like retinotopy, where the neural activity is really the same across depths, we have a knob that we can turn during the analysis. If we need more sensitivity, we can sample near to the pial surface, but if we're prioritizing neuronal specificity and spatial accuracy, we should sample near to the white matter surface. So it appears possible to use fMRI to achieve high resolution. And we expect to be able to achieve this specificity, in part because classical studies have shown us that the cortical columns can be resolved with hemodynamics-based intrinsic signal optical recording. So here one can clearly see columnar systems features well below 1 millimeter in size, for example, the iso-orientation domains in the middle here, and the classic ocular dominance column or zebra stripe pattern shown on the right. So while this method only sees intravascular signals and it's limited to the superficial cortical layers, it's still hemodynamic and therefore concretely demonstrates that the biological resolution is high. So if we can achieve small enough voxels, we expect to see columnar systems using human fMRI as well. And indeed we can. Evidence of ocular dominance columns has been seen in humans for about 25 years, but perhaps the best known example is from Issa Yacoub, who showed that spin echo BOLD, which is less sensitive to large veins compared to gradient echo BOLD, can be used to resolve this ODC zebra stripe pattern in humans. And so we later demonstrated that it's possible to image this ODC pattern with gradient equibold over a large region of V1 using our anatomically informed sampling approach. So here, our isotropic high-resolution acquisition and our surface-based analysis allowed us to avoid those pial veins and see this pattern over a large region of folded cortex. We're no longer restricted to locally flat patches of cortex as in previous studies. So this anatomically informed sampling provides a huge advantage, since gradient equibold has higher sensitivity than spin equibold, even for voxels that only contain capillaries. And so this is important because the difference in signal level in a given ODC is really tiny. And so here we need all of the sensitivity that we can get. And so we've also used this approach to look at columnar systems outside of V1, such as the V2 thick-thin pale stripe system. And in this case, I'm showing only the color-selective thin stripes. You can see these columnar stripes extending perpendicularly from the V1, V2 border, here in the inflated representation on the left, and in the flattened representation on the right. Now, if we sample this activation in the superficial gray matter near to the pial veins, we cannot resolve these stripes. But as we sample deeper, closer to the white matter, the columnar pattern becomes more distinct. And so these V2 stripes have now been seen by several groups, with this especially clear example from the Max Planck group in Leipzig, again using gradient equibold sampling away from the pial surface. Okay, so now I'd like to shift gears and consider other ways in which the vascular anatomy affect our fMRI signals, and in this case, the orientation dependence of BOLD fMRI. Now, this next section is work by Olivia Wiesmann when she was in my lab. So as our voxels get smaller, we have to be mindful of the influence of vascular geometry on BOLD. As I mentioned before, the cortical vasculature follows a strict geometry, and the large pial vessels lie tangentially on the surface, and the diving arterioles and ascending venules run perpendicularly to the surface. So this means that there's a coupling between the cortical geometry and the vascular geometry. Now, it's well known classically that the BOLD signal exhibits a dependence on vessel orientation relative to the direction of the magnetic field, or B0, as shown by this equation here. So as we go to higher field strengths, most of the BOLD signal is extravascular, and so this orientation effect can be summarized by a simple sine-squared relationship. So what this means is that when the vessel is perpendicular to B0, we see a strong extravascular field offset. However, if the vessel is parallel to B0, the extravascular field vanishes. This means that veins that run parallel to the magnetic field are essentially invisible to our fMRI measurements. So while this effect has been known for decades, it's usually neglected, perhaps because for large voxels, one can assume a random distribution of orientations. And so although the orientation dependence of a single vessel is well known, work by Louis Gagnon and David Boas's lab, using microvascular reconstructions from mouse cortex, which I'll come back to later, quantified through simulations the orientation effect expected from a voxel containing realistic microvascular anatomy. And so while there's no surprise that some effect exists, their simulations predicted a huge 40% effect. So that is cortical locations where the surface normal is parallel to B0, such that the large veins are perpendicular. We'd expect a BOLD signal change that's 40% greater compared to locations where the cortex is perpendicular to B0. So this was somewhat surprising, and so they went down to the scanner and by inducing a whole brain activation using a CO2 delivery, demonstrated that this effect could be observed in conventional BOLD fMRI data. And so we wanted to see whether this effect could be detected in resting state fMRI data and whether it varied across cortical depths. Indeed, in our high resolution 7T data, we saw that the resting state BOLD fluctuation amplitudes that are used for estimating functional conductivity varied with orientation, and this effect changes across cortical depths. So here we see from voxels sampling near to the pial surface, as shown in the yellow mask on the right, that the resting state BOLD fluctuations were 70% larger in cortical locations where the cortex is parallel to B0 compared to locations where it's perpendicular. So this is a strong effect. And as we considered voxels deeper into the cortex, further away from those large pial veins, we saw that the orientation effect started to melt away, as expected. And the effect nearly vanishes for depths near to the white matter, only about a 10% effect there. But then to our surprise, as we went deeper, looking at voxels closer to the white matter interface and pushing into the superficial white matter, we saw that the orientation effect started to rebound. And so to be clear, this observed rebound is consistent with an influence of some vessels that run tangential to the cortical surface, but here it's seen in voxels sampling very far away from the large pial vessels. And so at first we thought this was a mistake, but then Olivia remembered the classical descriptions of large veins within the superficial white matter running tangential to and kind of hugging the gray-white border indicated by these purple arrows. We think that it is these vessels that are causing this rebound of the orientation effect that we see at the white matter border. And so we're seeing this orientation effect also in high-quality modern data sets with more conventional voxel sizes, such as that provided by the Human Connectome Project, which shows about a 20% orientation effect in their 3T resting state fMRI data as well. So this suggests that it may be a little less likely to see functional connectivity in cortical locations that are perpendicular to B0, but this should be straightforward to correct. Now, these orientation effects are driven by a coupling between the pial vasculature and the cortical gray matter geometry. However, similar couplings exist elsewhere in the brain, such as a potential coupling between vessels and tracts of the white matter. I say potential because less is known about the quantitative relationship between white matter vessels and tracts. So here I'm showing a classic microradiograph from the 1960s of the white matter vasculature after ink injection on the left, next to diffusion tractography on the right. And you can appreciate the qualitative similarity between the vessels and tracts. So we wanted to investigate whether a similar orientation effect could be observed in BOLD fMRI in the white matter. Now, this is motivated by reports of patterned BOLD fMRI in the white matter that appear to follow the tract geometry. And here are a few examples. So it's unclear what the basis of these remarkable findings is, and perhaps, like the gray matter, the vasculature in the white matter is matched to the surrounding anatomy. So we sought to test whether we could detect influences of the white matter vasculature and its geometry on white matter BOLD, and to see whether this white matter vasculature really does match up with the tract geometry. If it does, not only would this help with the interpretation of white matter fMRI, but it would also show us a conspicuous relationship between large-scale vascular and tissue anatomies. So we checked for a similar orientation effect instead of looking for changes in BOLD amplitude with the orientation relative to the normal of the cortical gray matter surface. Here we looked for changes in BOLD amplitude with the orientation relative to the tangent of the white matter tract, estimated from diffusion tractography. So here's a movie summarizing the results. We find that many, but not all bundles exhibit an orientation effect in the BOLD fluctuation amplitudes. So first, on the left, we saw that all tracts exhibit an orientation effect in the baseline of the BOLD signal, which is expected because the tissue-tissue star value is dependent on the orientation of the myelin of the tract. But on the right, more interestingly, you can see that the orientation dependencies of the BOLD signal fluctuations, or dynamics, are more heterogeneous across the bundles. Many show the expected negative sine-squared orientation trend, which is consistent with dynamic BOLD effects in vessels that are aligned to or parallel to the tracts, and we see up to a 20% effect. But in other bundles, we did not see an orientation effect in the BOLD fluctuations. So this tells us that the blood vessels do appear to run alongside many of the white matter tracts, which would cause BOLD responses to vanish in any locations where the tracts are parallel to B0. But why do we see a clear orientation effect in the BOLD dynamics in some tracts and not others? So to try to understand this, we sought to directly image the white matter vessels along with the white matter tracts. So this is work from Mike Bernier while he was in the lab. So to achieve this goal, Mike used the blood pool contrast agent Ferumoxytol to enhance the deep white matter vessels. This was needed because it's otherwise very difficult to image these vessels due to their small size, and only a handful of white matter vessels are typically detectable, which you can see on the left. Now, Ferumoxytol enhances both arteries and veins. In our data on the right, you can see a huge improvement in the depiction of white matter vessels. So with this, we were able to examine the relationship between white matter vessels and white matter tracts imaged concurrently within subjects. So here is an example comparing Mike's vascular segmentation with the tractography estimate for the occipito-pontine tract, where we can see a decent correspondence between vessel and tract geometry. So here are a few other examples of bundles that exhibit a relationship between white matter vessels and white matter tracts, seen in other subjects, this time using an atlas of the white matter bundles. Indeed, we see that many bundles appear to have large vessels clearly running alongside them. But not all tracts are vascularized. However, we compared the results across these two studies, and while there's a lot of information in this plot, the main takeaway is that the bundles with high vascular density, seen by Mike, are also the bundles with a strong BOLD orientation effect, seen by Olivia. Now recall that this comparison is across very different data sets, including our Ferumoxytol data and the HCP fMRI data, and we do see some consistency. So this increases our confidence in our finding that some tracts are just more vascularized than others, and in those vascularized bundles, the vessels do appear to run parallel to the tracts. So just to close this section, I presented both indirect and direct evidence that white matter vessels often run alongside the white matter tracts. So how do we interpret this in the light of recent white matter fMRI studies? It's difficult since there may simply be a relationship between white matter vascular anatomy and functional anatomy at large scales, just like in the gray matter. And the BOLD signal changes in the white matter could still reflect local neuronal activity there. But like any BOLD observations, there are biases imparted by the vascular geometry. Okay, so now that we've seen how both small-scale and large-scale vascular organization are matched to the surrounding anatomy, I'd like to talk about how large-scale vascular organization may affect resting state fMRI data. So this is work by Jingyuan Chen, shown here, who is interested in how slow changes in systemic brain physiology affect the spatial patterns of BOLD fluctuations. Now, previous studies, especially this early study from Rasmus Byrne, have shown that large components of the BOLD response are driven by respiration volume, heart rate variability, end-tidal CO2 changes, as well as other systemic physiological changes such as blood pressure dynamics. Now, often physiological components of these resting state fMRI signals are recognized through their characteristic spatial patterns. However, few studies have really examined how the regional heterogeneity of these physiological responses might influence functional connectivity estimates. Now, physiological fluctuations have been shown to be localized to large blood vessels, as shown here, and so the vascular anatomy appears to shape these physiological patterns. Now, other components, however, are thought to be global and spatially invariant, therefore are unlikely to influence functional connectivity estimates. Now, conventionally, the BOLD response to these physiological signals is considered to be globally similar and is modeled by a single canonical response function, including the responses to cardiac, respiratory, and CO2 changes as shown here. However, we might expect these responses to really vary across brain regions because, for example, it's well known that the blood arrival time is inconsistent across the brain and because, of course, of the heterogeneous vascular density and anatomy. So therefore, we reason that these physiological responses may differ both in timing and in amplitude across brain regions. And so to test this, we deconvolved the slow changes of external physiological recordings from the resting state BOLD time course at each voxel. We leveraged the large number of subjects provided by the HCP dataset and estimated the responses after correcting for instantaneous physiological noise, in this case using retro-ICOR. And so here are the results of this analysis based on, again, the recording of respiration. So this is essentially a per voxel BOLD respiratory response function, or you can think of it as a 4D respiratory response function that varies over space and time. And so the average time course shown above exhibited strong differences across the brain, and below you can see the timing and amplitude of the response varied substantially across the brain. And so we then performed a data-driven clustering analysis on this respiratory response function to identify regions with similar responses. And indeed, we pulled out strong regional differences in this respiratory response function across the brain. So below are the mean response functions within each cluster. So note that some of these clusterings resemble patterns we might see in functional connectivity analysis that could be easily mistaken for neuronal networks. So this suggests that correlation analyses may detect patterns that are purely driven by similarities in this respiration response function, which we might call respiratory networks. And so we performed the same deconvolution on slow changes of the heart rate recording to estimate per voxel cardiac response functions. Again, the patterns were largely consistent across subjects and showing a spatially varying amplitude and timing of this 4D BOLD cardiac response function. And again, data-driven clustering analysis revealed several clusterings that resemble neuronal networks, and thus could potentially be mistaken for neuronal networks in standard functional connectivity analyses. And so note that these patterns persist even after physiological denoising, in this case using ICA fix. Now, these results here were not included in our original article, but since many folks were interested to see the effects of denoising, Jingyuan is presenting this follow-up in an abstract at this meeting, so please check it out if you'd like to learn more. Okay, so now to test for similarity between physiological networks and conventional resting state networks, we compared this respiratory connectivity with spontaneous functional connectivity. We did this by generating two data sets. The first was a synthesized data set containing only respiration-driven dynamics, which we labeled as RESP. So these synthetic data are pure physiological noise. The second data set consisted of resting state BOLD after careful removal of all physiological fluctuations, including those used to derive our response functions, which is basically our best estimate of BOLD data that's at least mostly driven by neuronal fluctuations. And so we then performed a seed-based functional connectivity for each of these two data sets. We saw that when using a seed from the visual network, the correlations observed in the synthetic data, again, containing only respiration-driven fluctuations or pure physiological noise, closely resembled the correlations that we saw in the denoised data, containing only or mostly neuronal fluctuations. We saw a similar resemblance between the two in the connectivity estimated from the dorsal PCC as a seed. Now, a somewhat similar observation was made by Yunji Tong and Blaise Frederick, who also generated synthetic data sets, in their case based on spatially varying blood arrival times. And so what does this mean? So the physiological networks seem to mimic the neuronal networks. Now, while this observation appears robust, the interpretation right now is unclear. It may be tempting to conclude that some aspects of functional connectivity could be a physiological artifact, but I don't think that this is the case. In fact, the lesson here may be more subtle than this. It may just be that there is some relationship between the regional vascular anatomy or physiology with the underlying functional organization. And recent intriguing data from Molly Bright and Kevin Murphy further suggests that the vascular anatomy in some way reflects the underlying neuronal function, which is a cool hypothesis and consistent with our results. So in their study, Molly and colleagues collected task-driven BOLD fMRI data using stimuli to generate neural activation, plus they had the subject breathe in CO2, again, a potent vasodilator, to generate vascular activation. And using a data-driven ICA-based approach, they identified 3 well-known large-scale networks, clearly driven by the neuronal activation. But what was fascinating is that they also identified for each of these networks a secondary network that was sort of spatially overlapping, but whose BOLD response was strongly correlated with the vascular stimulus timing. So this plot shows that for each of these 3 network pairs, the neural network is mainly driven by the neural stimuli, while the associated vascular network is driven only by the vascular stimulus. So both of our studies demonstrate physiological networks in the fMRI data that seem to generate network-like patterns that mimic neuronal networks. But Molly's study shows that these neural networks and the physiological networks coexist and are strongly resembling one another. So this teaches us that at a large scale, the vascular architecture reflects the functional architecture. Okay, so next I'm going to shift gears again and talk about biophysical modeling of the fMRI response to try to better understand these vascular influences. Now, the biophysical models that I'll be discussing are based on both realistic microvascular anatomy and dynamics. Now, this BOLD modeling approach that I briefly mentioned earlier was pioneered by David Boas and his group. And so we call this approach the Vascular Anatomical Network, or VAN modeling approach, as it differs from other BOLD modeling approaches, such as the balloon model. So in a bit more detail, in this approach, we utilize a full network from one location of cortex, consisting of a handful of input arteries and output veins, and a couple thousand capillary segments. So with this, we simulate the full spatiotemporal dynamics of the hemodynamic response. Now, the input to the model is a time trace of the feeding artery diameter changes, which reflect the vascular response to neural activity. We use computational fluid dynamics to simulate local blood pressure, flow, and volume changes, blood oxygen advection, and oxygen diffusion into the tissue. We then compute the magnetic field offsets in and around the vessels, and finally the T2 or T2 star weighted signal, which gives us the BOLD response. And so these models differ from previous approaches because the vascular anatomy is connected into a single network, which allows us to simulate the dynamics of the microvascular response, as shown in this animation, and to understand how both the geometry and topology of the microvasculature can shape the BOLD fMRI signal. And so we can use a synthetic vascular network with this approach and parametrically vary features of the microvascular anatomy to see how it influences the BOLD signal. So if we start with maximal capillary density in layer 4, which can be seen in this rendering and in the histogram below, our simulation shows that the resulting BOLD response, the amplitude peaks at the pial surface, but it exhibits a bump or local peak in layer 4, even if we assume that the neural activity is the same across all cortical depths. So from there, we can synthesize additional vascular models in which we've moved the peak capillary density to the upper half of the cortex, to the middle third, to the lower half, and in each case, the BOLD peak follows the capillary density peak. This shows us that in addition to macrovascular biases, such as the large BOLD signal seen at the pial surface, there may be microvascular biases as well, where the BOLD signal is largest in regions of high capillary density. So what does this mean for the interpretation of high-resolution or laminar fMRI using BOLD? Or what do we conclude if we see a local peak in our BOLD data in layer 4? It's tricky. Again, because the microvascular architecture reflects the functional architecture. And there seems to be a peak capillary density in layer 4 for a good reason, which is that there's stronger neural activity there and higher metabolic needs. So this points to a key challenge in interpreting these data. And so as I mentioned earlier, the vascular architecture of the cortex follows the neuronal architecture. However, the vascular layers may not be fully independent from one another. Now, many models of the intracortical vasculature assume there's a number of distinct capillary beds that each supply a particular cortical depth. So as you can see in the schematic, the capillaries, shown in purple, run largely parallel to the cortex and do not cross the layer boundaries. Now, while this is a reasonable simplification, capillaries are less ordered than this. And so in this reconstruction on the left, you can see that these capillaries in blue often run perpendicular to the cortex, cutting across several layers. We call these trans-laminar capillaries, or capillaries that traverse several layers. So I've gone in and added a few of these to the schematic on the right. Now, the existence of these trans-laminar capillaries means that even if we can sensitize our fMRI measurement to hemodynamic changes from small vessels only, there may still be a spreading or coupling of the hemodynamic response across layers. So to understand the impact of translaminar capillaries on the fMRI signal, we've been using Van models to simulate microvascular point spread functions. So this is work by Grant Hartung and Jörg Vanmöller, and Grant is here at OHBM presenting this work, so I encourage you to go check out his poster. So here they're using reconstructions of the full microvasculature from one location in the mouse cortex and modeling the spread of blood flow and oxygenation that arises from the smallest possible activation of the vasculature. So to motivate their simulation approach, the vessel segments closest to neurons that are universally accepted to actively regulate blood flow are the pre-capillary arterioles, shown here in yellow. Now, for this reason, in our simulations, these pre-capillary arterioles are the sites of the control structures, and by definition, each capillary network is fed by a pre-capillary arteriole. So our goal is to calculate how the blood flow spreads when we activate a single pre-capillary arteriole, and there's several of them across cortical depths. So the idea is that if we activate them one at a time to increase the blood flow and oxygenation into the downstream capillary network that they feed, then quantify the spread of the response across depths to measure the point spread function of the activation. Now, the extent to which multiple activated pre-capillary arterioles result in overlapping blood oxygenation responses will introduce ambiguity when trying to relate the measured BOLD signal back to the activated cortical layer or layers. And so to quantify the impact of these translaminar capillaries, we used a mouse Van model from mouse area S1 that spans the entire cortical thickness from the pial to the white matter borders. So here you can see an example capillary traversing a long distance along the cortical depths, similar to what I drew on the schematic a few slides back. So this is a nice example of a clear translaminar capillary. So here's another example of a capillary, this time one that is roughly parallel to the cortical layers. We might call this an interlaminar capillary. However, it seems possible that most of the capillaries are translaminar. So here is one example of a pre-capillary arteriole. In our simulations, we activate one arteriole at a time and simulate the resulting blood flow and oxygenation changes occurring in the downstream capillaries. And here's the corresponding simulated BOLD response from simulating this one arterial dilation. We repeat this process for each arteriole in the Van model, activating them one at a time, and then quantify the resulting spread of the blood oxygenation response. So we found that by activating about 550 pre-capillary arterioles in this model, the spread of the blood oxygenation perpendicular to the cortex or across depths was about 400 microns on average, with slight differences seen at the very top and the very bottom of the cortex as shown here. And the results were similar across the 4 Van models that we tested, and I encourage you to visit Grant's poster to see more of the simulations. So now this average spread corresponds to 40% of the cortical thickness, and so it's equivalent to roughly 3 cytoarchitectonic layers in S1. So this suggests that if the pre-capillary arterioles are the only control structures that regulate blood flow in the cortex, perhaps 2 to 3 layers could be resolvable with BOLD fMRI, provided we have small enough voxels, and if we use techniques like pure spinnacle BOLD that are sensitive only to the capillaries. Now, pericytes may help shape the microvascular response, and we're just beginning to consider whether they can tighten it. Now, knowledge of this microvascular point spread could potentially help infer the locus of neural activity for measured fMRI data, and with clever experimental design and analysis informed by these simulations, we should be able to better localize the activated layer or layers. Now, the vascular architecture is quite different between mouse and human. However, we have seen some evidence for translaminar capillaries in humans, so we think that they are relevant to the biological resolution of human fMRI as well. Okay, so finally, I'd like to wrap up by considering the biological temporal resolution of fMRI. So this is work led by Laura Lewis, who wanted to see whether we could track fast BOLD responses to rapidly oscillating visual stimuli. Now, we're mainly interested in endogenous oscillations, but in this case, we induced neural oscillations with a known waveform and frequency using visual stimulation consisting of a simple flashing checkerboard whose luminance contrast was modulated at several temporal frequencies of interest. So to track these BOLD responses, we acquired fast fMRI data with a short temporal sampling interval of around 250 milliseconds. So if we consider the predicted BOLD responses to the stimulus using a canonical hemodynamic response function, we see that at low frequencies, the predicted BOLD response clearly tracks the stimulus. However, as we move to higher and higher oscillation frequencies, to the point that the oscillation period is short compared to the temporal width of the hemodynamic response function, we see that after an initial transient, the BOLD response oscillates around a new baseline or plateau, and the amplitude of the oscillations around this plateau gets smaller and smaller with higher frequencies, exponentially, but never quite reaches zero. So the question is, what oscillation frequency might we be able to detect with our BOLD fMRI data? So in these trial-triggered averages, you can see that the BOLD signal was able to track both 0.2 hertz oscillations as well as oscillations as high as 0.75 hertz at 7 Tesla. This is a far higher frequency than what we had imagined that we could detect. Note the vertical axes on these plots. The BOLD response to the 0.75 hertz stimulus was tiny, but still detectable, yet it was still 10 times larger than predicted using the canonical hemodynamic response function. So we know that the HRF can be nonlinear when using short duration stimuli, such that larger amplitudes are observed relative to those predicted from a linear model. So there's some potential explanation for this observation. Now, to test whether we could detect even faster oscillations with higher SNR, we attempted, just in 2 subjects, to see whether we could go up to 1 hertz at 9.4 Tesla in collaboration with Klaus Scheffler at the Max Planck Institute in Tübingen. So this turned out to be difficult since 1 hertz is right up against the cardiac frequency, but with some careful physiological denoising, we were able to track 1 hertz oscillations in the visual cortex in both subjects, even though the linear model predicts these to be essentially undetectable, suggesting that maybe, with even higher sensitivity, we could detect even faster frequencies with BOLD fMRI. And finally, similar oscillations have been reported in motor cortex and in somatosensory cortex in this collaborative work with Shota Hodono and Martijn Kloos at the University of Queensland. Here, using a data glove that records hand motion during MRI scanning, they also measured BOLD responses tracking up to 0.5 hertz activation with amplitudes far higher than the predicted in the canonical model. They also examined the response across cortical depths and found that at low frequencies, the BOLD response is largest near the pial surface, but at higher frequencies, including 0.5 hertz, the depth profile flattens, suggesting that large draining veins at the pial surface can't keep up, but the small vessels within the parenchyma are relatively faster, suggesting that these microvessels may not only exhibit improved spatial specificity, but perhaps improved temporal specificity as well. So to summarize, we have shown that the BOLD fMRI can track oscillatory neural activity up to 0.75 hertz and perhaps up to 1 hertz. So this suggests that fMRI signal can reflect meaningful neuronal dynamics approaching the timescales of many cognitive processes. However, this steady state BOLD response generated by our stimulus, which sort of gently perturbs the hemodynamics around a new plateau, may actually provide faster temporal hemodynamics needed to track these oscillations. Now, these data also imply that the canonical HRF, derived from long stimulus blocks like intense 24-second blocks of flashing checkerboards, may not be appropriate for capturing the BOLD response to subtle or ongoing cognitive processes or more naturalistic stimuli. And so we're currently beginning to use our biophysical modeling framework to better understand these fast hemodynamics and the nonlinearities. Okay, so to conclude, I've described how anatomically informed sampling of BOLD fMRI data can in some cases improve neuronal specificity. I've also shown how vascular geometry is coupled to tissue geometry in gray matter and white matter, and how this imparts an orientation bias in BOLD fMRI. I've shown that at large spatial scales, the vascular or physiological networks resemble or perhaps accompany neuronal networks. I've also shown how at small spatial scales, biophysical models can provide insight into microvascular influences on the fMRI signal and how it can perhaps help us to understand the ultimate biological resolution of fMRI. Now, as I just said, the BOLD response tracks high temporal frequency activation and exhibits high temporal specificity, and we were able to detect faster responses than we had imagined. And finally, to try to weave these topics together, I'd say that we're learning more about how the vasculature shapes or influences the fMRI signals, and while the ultimate biological resolution of fMRI is not known, there is a limit, but we haven't reached it yet. And so while I'm happy to see this trend towards fMRI acquisition and analysis methods that achieve high imaging resolution and techniques for providing microvascular specificity, these can be combined with perhaps vascular imaging data and models of vascular physiology to help improve interpretability. So today, fMRI has untapped potential. And by appreciating these vascular influences, we can extract more meaningful information from our data. And so with that, I'd like to thank the following people, Laura, Jingyuan, Olivia, Mike, Grant, Shaheen, and Shota for contributing slides, my lab, as well as the MR Pigs, our funding sources for their generous support, and thank you for your attention.